Hi everybody, here is the summary of our letter that has been accepted in the Allergy journal and entitled Induction of Immunosuppressive CD39 molecule but not the trafficking CCR4 on Tregs in dual responders mite allergic asthmatics. Animal studies showed that Tregs play an important role in the control of the asthmatic response. In human, it has been suggested that Tregs were deficient either in function or in number in allergic asthma, and our aim was to characterize Treg response after DPT challenge in mite allergic asthmatics. 17 GNL level 1 well-controlled mite asthmatic subjects were included and exposed to placebo at visit 1 and to DPT at visit 2 in the Aliatec environmental exposure chamber. The early and the late asthmatic response were assessed by the drop of FEV1, defined as a 20% drop in FEV1 for early responders and a 15% drop in FEV1 for late asthmatic responders. Blood samples were obtained before and 6 hours after exposure. CD4, Tregs, defined as CD4 plus CD25 high FOXP3 positive, and surface molecules implicated in the function, ICOS, PD1, and CD39, and in the lung recruitment, CCR4, of Tregs were measured by flow cytometry. Asthmatic subjects were categorized in 2 groups according to the asthmatic response, early and dual response. 13 subjects exhibited a dual response and 4 an early response after DPT exposure, but not to the placebo. At baseline, discriminant variables between dual and early responders groups were Treg CCR4, CD4 CCR4, the ratio between the Treg CCR4 and CD4 CCR4, and the sensitization against the DPT, either measured by skin prick test or specific IgE. After DPT challenge, we observed an increase in CCR4 expression on CD4 cells, as well as an increase in IL-2 and IL-5 levels compared with placebo in dual responders, confirming the inflammatory response occurring during the late asthmatic response. Among the immunosuppressive variables, expression of CD39 increased in dual responders compared with placebo, but no significant changes in Treg, IL-10 and TGF-beta levels, nor in the expression of CCR4 on Tregs were observed. Taken together, our results suggest that dual responder subjects exhibited a higher T2 profile at baseline as well as after allergen exposure, characterized by lung-homing T-cell molecules, IL-2 and IL-5 cytokines compared with early responder subjects on one hand. On the other hand, DPT exposure induced the expression of CD39 on Treg cells, which has been implicated in the suppressive function of Tregs, but not of the recruitment CCR4 receptor. This observation may explain the lack of Treg recruitment to the lung during the late asthmatic response that has been previously reported. We thank you for your attention and your interest in our work.